Hello, my name is Anna. I will present a group research on 3 mutants on chromosome 20. The first part of our project is gene prioritization. We focus on Carl's chromosome 20 and prioritize genes based on the number of SNPs or point mutations in the exon region of protein coding genes. As a result, the top 10 protein coding genes, including 13 transcripts with the highest number of mutations in their exons, were selected for downstream analysis. The second part of our project is protein structure analysis. The analysis process involved 4 steps. First, the mutant sequence were constructed by altering the transcript sequence from the ensemble database. Splicing and translation were subsequently performed. Second, the mutant protein structure was predicted by running AlphaFold in Google Colab. 5 unrelaxed structures were generated for each mutant sequence, and the top ranking prediction was chosen for downstream analysis. Third, the local distance difference test, also known as the LDDT scores, were calculated for the wild-type and mutant structures to understand how much a mutation affects the global structure, where a value close to 1 indicates high similarity. And finally, the structures were visualized in PyMo, and the SNPs were highlighted to study areas of the protein structure that are affected. Based on the availability of the wild-type structures, we narrowed down our studies to 3 proteins and their mutants. MTG2 is a protein involved in the regulation of the mitochondrial ribosome assembly and of translational activity. In the wild-type, residue 278, threonine, is on the flexible loop, but the mutated residue methionine constitutes the alpha helix. The mutation was, however, benign. The LDDT score is consistent with the visualization, where we see slight structural alterations. JPH2 is a cardiac structure protein. Although pathogenic mutations in the encoding gene are associated with cardiac diseases, Carl's mutation at residue 261 from valine to isoleucine is benign and does not affect the alpha helical structure. The LDDT score is consistent with the visualization, where we see the structure remaining mostly unchanged. Kia 1755 is an uncharacterized protein. We noticed 3 mutations. However, they did not alter the protein's secondary structure significantly and are not associated with any reported disease implications. The LDDT score is consistent with the visualization, where we see slight structure changes at the loop regions. So we'd like to conclude that all mutations that we studied on Carl's chromosome 20 are potentially benign. Our code and data are all available on Github. Thanks for watching.